So we are going to go over DNA. This is the last bit of stuff we got to go over for AP biology. DNA, how it's replicated, transcribed, translated, we've briefly talked about, but we're going to go into way more detail with it. So a little bit on the history of DNA. So there's a lot of scientists that help contribute to what we know today as the DNA replication process, transcription, translation. So these are some of the names. We'll go a little bit more in detail with all of them. So we have this dude, T.H. Morgan. He found out that chromosomes are related to your phenotype. Okay, so he worked with those same fruit flies from the last lecture, and he associated the phenotype with specific chromosomes or sex-linked traits. Okay, so we get certain physical traits from either X or Y. So his conclusions are that the genes are physically on the chromosomes, but he needed to know, is it the protein or the DNA of the chromosomes that are the genes? So initially, he thought proteins were the chromosomes that are on the genes, and he thought that those were the genetic material. Now, why do you think he thought that? Oh, because proteins are freaking everywhere. Also, the proteins are wrapped around the DNA, okay, for those histones. So, Frederick Griffith used a certain form of pneumonia bacteria that he was working with to find a cure for pneumonia. So he had harmless live bacteria mixed with heat-killed pathogenic bacteria, which caused fatal disease in mice. So a substance passed from the dead bacteria to the live bacteria to change their phenotype. So he noticed that it was called the transforming principle. So here, if you had, the original live bacteria mixed with the killed bacteria, then that's when you would get, it would transform the phenotype to where you would end up getting a dead mouse. So then if you put the live non-pathogenic strain of bacteria, the mice would live. If you put the heat-killed pathogenic bacteria, the mice would live. But if you mixed the two, you'd die. So there was a transformation, meaning a change in the phenotype. Something in the heat-killed bacteria could still transmit the disease-causing properties. So this is just what he looked at specifically. So then we had these dudes, Avery, McCarty, and McLeod, who purified both DNA and proteins separately from that pneumonia bacteria. They said this will transform they wanted to know, will this transform the non-pathogenic bacteria? So they injected the protein into the bacteria and had no effect. But then they injected the DNA into the bacteria, and that transformed the harmless bacteria into the virus bacteria. Okay, or the virulent bacteria. So they realized then that it was the DNA, not the protein, that was the genetic material. So this was the very, very first experimental evidence that DNA was the genetic material. So go ahead and pause the video, read through this question, and get your answer. You should get that your answer is A, DNA is the genetic material, okay, because we just went over their experiment. But then also reading through, you can see why it is that case. So now a confirmation of what DNA is. So Hershey and Chase, they're famous for a classic blender experiment. So they worked with a bacteriophage, which is a type of virus, and it's a virus that infects bacteria only. So they grew the bacteriophage virus in two different media, so two different methods, radioactively labeled with either sulfur 35 in their proteins or phosphate 32 in their DNA. So they infected the bacteria with these two different bacteriophages. This is Martha Chase and Alfred Hershey. These are the two that conducted this experiment. Yay, it's a girl. Okay, so what they did is here you can see protein coat that is labeled with sulfur 35 is red, and the DNA labeled with phosphate 32 is red, and then they injected them into the bacteria to kind of see what happens. So what they found out is that sulfur 35 radioactivity is found in the medium, and then the phosphate 32 radioactivity is found in the bacterial cells. So this is kind of an overview of what they ended up doing. So they mixed the different viruses, the radioactive viruses, to infect the cells, okay? And then they mix them in the blender, okay, and centrifuge to kind of see the DNA at the bottom, okay? And then they did the same thing for phosphate. So what they found out is that for the sulfur phage, radioactive protein stayed in the supernatant. So the viral protein did not enter the bacteria. Supernatant is just like the extracellular material. For the phosphate, the radioactive DNA stayed in the pellet, so the DNA did enter the bacteria. So this confirms that DNA is a transforming factor to kind of prove once again that DNA or nucleic acids are the molecules of heredity, not proteins. So then we had Erwin Shargriff, who came up with Shargriff's rules for the DNA composition. He's the one that found out that DNA composition obviously is going to vary from species to species, but all 4 bases are not in equal quantity. But the bases present in characteristic ratio in human is this. So you can see A to T are pretty similar in the percentages, and G and C are similar in the percentages. That's because A bonds with T and G bonds with C. A and G are your purines, T and C are your pyrimidines. Okay, now to get the actual structure, that double helix structure of DNA, James Watson and Francis Crick take credit for it. But Rosalind Franklin was very, very important in this. So this is the structure of DNA. Obviously, it is double helix. So this is Watson and Crick. Okay. If you want to read more about it, you can click on this article. They're very famous, obviously, because they presented this, but Rosalind Franklin was actually the one who did the X-ray diffraction and saw that DNA was the double helix. What ended up happening was that she kind of like left her work out. Watson and Crick found it, saw it, took credit for it, and so she's not known for finding it, but she is actually the one who found this X-ray picture of DNA having that double helix. Now, because she was involved in a lot of the like X-ray and ultraviolet radioactive activities, she did end up developing cancer and dying. So it, kind of didn't really help that she wasn't alive to kind of fight them off type of thing. But Rosalind Franklin did actually find out that it was double helix first. So once again, yay, women, woo. Okay, so DNA structure. We can see that we have the sugar phosphate backbone. In DNA, the sugar is deoxyribose. RNA, it is just ribose, okay, because DNA stands for deoxyribonucleic acid. Okay, so you have your sugar phosphate backbone, and then in the middle you have your nitrogenous bases. What's binding your nitrogenous bases together are hydrogen bonds. Okay, so the little circle on the side is your phosphate molecule, and then the little house in the middle is your sugar. Okay, and in the middle you have your nitrogen bases, so your nucleotide bases, bonding through hydrogen. So this is kind of to give you an overview. You see DNA is this ladder type structure, and then here's kind of the closer up breakdown of the actual molecules themselves. So here's some more ways that you can see how DNA is bonded. Watson and Crick said, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So they started theorizing that, hey, DNA can copy itself. Wouldn't that be awesome? So we'll get into that. So the actual bonding within DNA, the two different strands are anti-parallel, which means they're parallel, but they run opposite. So there's a 5' to a 3' direction, and then the opposite matching side would be 3' to 5'. So you can see that in between the sugar and the phosphate, you have phosphodiester bonds that are covalent, means that they're sharing. Okay, and once again, in the middle, you have hydrogen bonds. Now remember, hydrogen bonds are very, very weak. Okay, so between the bases, A and T are paired with 2 hydrogen bonds, okay, but C and G are paired with 3 hydrogen bonds. So your purines are adenine and guanine, your pyrimidines are thymine and cytosine. Okay, so for pairing, you have A to T, which is 2 hydrogen bonds, C to G, which is 3 hydrogen bonds. This is to show you how the hydrogens are actually bonded between these base pairs. Some more ways to see them. Now go ahead, pause, read the question. Remember, read the question first before looking at everything else, okay? Okay, you should have gotten B because A and T are pretty much the same in each sample, so obviously you know that they should bind together in DNA, okay? So this is what I was talking about with it being anti-parallel. You have one strand being a 5' to 3' direction, and the other strand is the opposite, so 3' to 5'. So nucleotides in the DNA backbone are bonded from phosphate to sugar between 3' and 5' carbons. What they mean by 3' and 5', the little dash next to the numbers, okay, stand for prime. So here you can see on this side it's a 3' and then a 5'. Okay, so anytime you see that hydroxyl, that's going to be 3'. So DNA molecule has a direction, and the complementary strand, so the matching strand, runs in the opposite direction. So DNA has sense and antisense regions. Antisense means the strand translated into mRNA for whenever we go through transcription. The sense is a possible role in the gene expression. So both sides of the DNA have sense and antisense regions. So it's just parts of the DNA that will be translated into RNA, or parts that are going to be used for actual gene expression. So some genes can be read in forward and reverse to produce different proteins. So this will kind of make more sense once we go over transcription and translation. So how is DNA copied? If you remember from mitosis, the very first phase of the cell cycle is called interphase. And during interphase, you have the S phase where DNA is replicated. That is the most important phase of interphase where DNA is actually replicated. So now we're going to go in depth how DNA is actually copied. So there are different theories of how DNA is replicated, okay? We have the conservative, semi-conservative, and then the dispersive. Now, the actual way that DNA is replicated is the semi-conservative replication, okay? So we want to conserve, the amount of energy and stuff that we do, but we don't need to conserve all of it. So Mailson and Stahl did a DNA replication experiment where they labeled the parent nucleotides in DNA strands with nitrogen 15, okay, and labeled the new nucleotides with a lighter one, which is nitrogen 14, okay. So this is the nitrogen 15 with the red, okay, so first round of replication, this is the conservative, nitrogen 15, nitrogen 14, okay. Semi-conservative, this is how it was set up. And then dispersive, this is how it was set up. So for the second round of replication, this is what ended up happening. Okay. So what they ended up actually finding is that the semi-conservative replication worked the best, okay? Because that's how you can actually see that DNA replicates best in that sense, because you can see that there was actual, replication occurring, whereas here there wasn't. All right, so I'm going to pause and then we will continue in the next one.